Hi, this is Mike Gilson. I'm a co-founder of the company VeraChem, and in this video, I want to introduce you to Mining Minima, a method for estimating protein-ligand binding affinities, or free energies, that I think offers a really nice balance of accuracy and computational speed, as well as VM2, a well-engineered and well-supported software package that runs the Mining Minima method. Just by way of background, VeraChem is a Maryland-based company that develops mostly physics-based software tools for computer-aided drug discovery. You can go to our website and learn more about the company and the software. But today I'll be focusing on VM2, as shown on the left here, and I'll also touch on QMVM2, a quantum method that I think has some unique capabilities. The structure of my talk is outlined here. I'll start off by introducing the theory of mining minima. So this is the statistical thermodynamics underlying the method. I'll talk a bit about how VM2 is implemented in our, how mining minima is implemented in our VM2 software. I'll then present benchmarks against experiment, looking at both accuracy and computational speed. And I'll close by talking about a couple more developmental topics, the use of chemical computing groups, MOE software, to run VM2 and analyze its outputs, thus giving VM2 a very powerful graphical user interface. And finally, as I just mentioned, the integration of quantum chemistry into mining minima in the QMVM2 software. So the basic paradigm we're working in is that of structure-based small molecule drug discovery. So you have a protein of known structure, as shown here, and you have a small molecule. You want to know how that compound will bind the protein. So that molecule could come from, medicinal chemistry design or computational design. It could come from a commercial catalog, or it could come from a generative model based on artificial intelligence. But the basic question is, is it likely to bind my targeted protein? And if so, how well? Or at least, is it better or worse than other compounds that we've looked at? So the basic theory of calculation of protein-ligand binding affinities is sort of underlies all methods in this field. So we have delta G bind equals negative RT ln of this quantity in brackets. C naught is the standard concentration. ZPL is what's called a configuration integral for the protein-ligand complex. And in the denominator, we have the configuration integrals of the protein and the ligand free in solution. And this basic theory can be used to derive, or at least motivate or rationalize a whole spectrum of methods, starting from docking methods, which are simple and fast and tend to be less accurate, all the way to probably the most detailed ones that are widely used, which are simulation-based free energy calculations, that are often called FEP these days, which are more detailed and slower, but more accurate. Mining minima is a method that really was designed to sort of sit in the middle of the spectrum of methods, offering greater accuracy than docking and greater speed than the simulation-based methods. In order to understand the method, I want to spend a little time talking about what a configuration integral is and how we use that idea. So the configuration integral of the protein or the ligand or the protein-ligand complex, so I'm taking out the subscript on the Z here, can be written in terms of a force field, U of R, which gives the potential energy of the system as a function of its internal coordinates, so R. So these are the coordinates of the protein or the complex or the ligand, along with an implicit solvent model, W, which gives the solvation free energy as a function of the same set of coordinates. And the basic property of this configuration integral is that the integrand is largest for low energy conformations. And so the more low energy conformations that the system has, the bigger the integral. And so going back to the prior equation, you can see that if the configuration integral for the complex is large relative to that for the protein times the ligand, you'll have a very positive large ratio here, and you'll end up getting a negative binding free energy, indicating that it's favorable, which makes sense because we're saying that the protein-ligand complex has more low energy or lower energy conformations. So the idea of mining minima is to directly estimate these configuration integrals. And we do it as a sum over the main local energy minima. So we're mining out the contributions of these minima to the configuration integral or to the free energy. And that's illustrated here for a very simplified energy model where there's only one dimension. Of course, a protein or a ligand has many dimensions, so we have some kind of complicated multidimensional energy surface. But the basic idea is shown here. If we have a coordinate over which the energy, so potential plus elevation free energy, varies and has these three minima, the biggest contributions to the configuration integral come from the deepest energy well 2, followed by the other two, 3 and 1. So we can estimate the overall configuration integral Z as a sum of local contributions from each of these wells. Within each well, we still have to evaluate this integral though, and we'll talk about how that works. But with this basic idea, the overall mining minima algorithm can be diagrammed as this loop which goes until the configuration integral is converged. And that's done separately for the protein, the ligand, and the complex. So we start with an initial local energy minimum, which is really just a conformation. So a conformation of the ligand, conformation of the protein, or of the complex with a certain ligand pose. We search for neighboring local energy minima, and during the search, we use a generalized Born surface area solvent model for speed. If we then discover that any of the new minima we found have already been counted, we delete those to avoid double counting. And then for the new ones that have been discovered, we compute the local configuration integral into integral Zi, where I is the index of the well. We also do a correction to the finite difference Poisson-Boltzmann surface area model, which tends to be a bit more accurate than generalized Born. So the contributions of these wells are added to the Z as a running sum, and if they didn't make much difference, then probably we've discovered all the low energy structures. It may be time to declare the run converged and report out the free energy, also known essentially as a chemical potential, for this particular species, the protein, the ligand, or the complex. If it's not converged, we go back, we search a local minima again, and we iterate to convergence. So how do we do the conformational search? We use a method called torque, which goes back a while. It's a fairly powerful method that, for those of you who are familiar with low mode search, it's essentially low mode search in torsional coordinates. So that's diagrammed here on a two-dimensional energy surface. We're going to start in this energy minimum and do a search that will hopefully lead us to this one. The basic idea is within this minimum, we compute the second derivative matrix of the energy, or also known as the Hessian matrix, in Cartesian coordinates, and we then transform that into internal so-called bond angle torsion coordinates. We then focus on the torsional subspace and diagonalize the matrix there, and we look for the lowest modes, the modes with the lowest eigenvalues. So those are the ones that correspond to the shallowest curvature as we go up away from the energy minimum, and those are the modes that will tend to lead us to mountain passes, basically, between peaks that we can then cross easily and fall into the next energy valley or energy well, as shown here. The idea is you want to distort along modes that will lead you up shallow slopes as opposed to steep ones. You don't want to go over mountain peaks, you want to go over passes. And so what happens is that the system is distorted along a low mode, or a combination of low modes, until the energy reaches some threshold. And then an energy minimization is started to drop you into the neighboring energy well. And then, of course, you have to filter out duplicates, and also occasionally, in case the stereochemistry has been damaged, that has to be filtered out. This is sort of a dramatic video showing somewhat extreme low mode distortions to give the idea. In blue or purple is the protein binding site with the rest of the protein removed. And in green, etc., is a ligand. And here we have a low mode search where you see the ligand and the protein moving in a concerted manner. And this is actually a rather powerful way of exploring various possible poses of the ligand and the binding site. What's not shown here is that after each of these distortions, there would normally be an energy minimization to relax down into the local energy minimum that's been found. This slide explains how we compute the configuration integrals and the local energy minima. We don't do a detailed integral as shown here. Instead, we do it an approximation that we call harmonic approximation / mode scanning, and I'll explain why. So let's say we want to compute the configuration integral for energy well I. Let's say it's a sort of an elliptical shape. We again compute the second derivative matrix of the energy plus salvation energy and get the principal axes of this elliptical shape. Really, now we have a Gaussian distribution for the Boltzmann factor. And then each mode, the contribution of each mode to the configuration integral can be estimated as a product of terms from each vibrational mode, if you will, in this harmonic energy well. Now, we're not really looking at vibrations, we're just looking at the curvatures because we don't need to worry about masses in classical statistical thermodynamics, but that's sort of the detail. So K1 is the force constant or eigenvalue for mode 1, K2 is the force constant or eigenvalue for mode 2. Each one makes a contribution to the overall configuration integral, and these are just multiplied out. Now, one thing we observed early on was that for the very lowest modes, the ones that are most easy to distort along, so the energy doesn't rise very quickly, sometimes the harmonic approximation is not very good. And you can see why in this diagram here. So this is energy as a function of some coordinate, some eigenmode. You see is that the energy function is initially very flat, and only as you move along it a ways does the energy rise sharply. If you compute or estimate the configuration integral based on the center here, right around the energy well, you're going to get a very, very low mode, which will give you a an excessively high estimate of the free energy. The harmonic approximation basically will think that this mode very slowly rises, that the energy rises very slowly along that mode, whereas in reality, at a certain point, it rises steeply. And so for the very lowest modes, we actually do a simple stepwise distortion along the mode, as shown here, compute the energy at each point, and use a quadrature method to get a more accurate estimate of the contribution of that mode to the overall configuration integral. And so what's shown here is that for the higher modes, again, we use the harmonic approximation. For the lowest modes, we use contributions from this special way of treating the soft modes, these are the ones that are mode scanned. Now, if we did a torque conformational search on the whole protein, it would unfold and not easily find its way back to the folded state. So that wouldn't be very productive for a calculation that's aimed at computing binding affinities. So in actual practice, what we do is we create a rigid outer framework around the binding site that the ligand and the binding site residues still feel. And then we treat the binding site residues and for the complex, also the ligand, as fully flexible and of course included in the energy function. And we call this the live set. The rigid set outside that is called the real set. And then to save time, one can actually discard parts of the protein beyond that. This method is implemented in the VM2 software package, which really now offers a lot of nice control capabilities and the ability to run in parallel in a variety of ways. So there's a Python workflow control, which takes care of system setup. It generates your input files, submits the jobs, and collects the results and summarizes and analyzes them. And then the workflow calls a compiled numerical core, which is a combination of C and Fortran code and allows for parallelism using MPI, OpenMP, and also GPUs. This core and the whole thing actually runs quite nicely on Amazon Web Services, which is great if you need some surge capacity at certain moments in a project, or if you don't want to manage a cluster yourself at all. And the method offers a binary restart capability. The workflow actually offers a number of interesting options that point to some of the special capabilities of the Mining Minima approach. So the approach calculations I've been talking about so far are so-called full VM2 calculations. So here you start with some pose, you then search around it. You do the same for the protein and the ligand. You do a comprehensive search of the energy surface as defined within your live set. And that can take around 10 minutes per ligand if you're using a GPU. But there are other scenarios which we think can also be really interesting and very efficient. One of them is to take a single pose, which may come from docking or from modeling on a graphical system like MOE, and just do what we call a free energy process or FE process on that single pose. So that can be fired off to VM2. We compute essentially the binding for energy for that docked pose rather than doing a full search. And that takes about a minute per ligand. And so this now becomes almost interactive and allows one to think about using a graphical user interface, sending a suggested ligand with a targeted pose off for calculation, getting some feedback, and editing interactively. In addition, we can do, or VM2 can do pure conformational searches on the binding side of the ligand without the free energy calculations. This works for proteins and ligands, host gas systems, and of course you can run it on the unbound ligand only if you're interested in the free ligand's conformational preferences, perhaps in relation to thinking about things like pre-organization. So some of our initial evaluations a number of years ago gave encouraging correlations with experiment. This is phosphodiesterase 10A. Here's calculation versus experiment. Next to our work from the lab of Professor Xia and Cheng at UC Riverside, again, encouraging correlations. Maybe most telling were these calculations from two different pharmaceutical companies, which were done entirely in their hands on receptors, which we actually don't know what they are. And again, without our sort of expert intervention, they got correlations with experiment on the order of 0.5, which again was quite encouraging, especially given the speed of the method. I'm going to show you another sort of anecdote that gives a sense for an additional capability of the overall package. Here we were looking at HIV protease inhibitors. This is a set of 38 compounds from University of Massachusetts, some of which had quite high affinities. And they have a common scaffold, so what they were varying was the R1, R2, and R3 groups, and they had measured affinities for all of these. So here's a sample, one of these compounds within HIV protease. What we did here was started with entirely unknown ligand conformations for almost all of them. So the ligands were input in 2 dimensions. There was one compound where there was a crystal structure with a pose for one of these ligands in the congeneric series. And what we did was use what we call our map snap tool, which looks at the bound ligand, looks at the ligand you want to fit into the binding site and compute the affinity, finds a common substructure, does a structural overlay, and then fits the rest of the ligand into the binding site to make a pose. So there, what you're doing is using the known conformation of the scaffold of one of the compounds in your series in the binding site to automatically map all the other ones in there and prepare for a nice uniform set of VM2 calculations. Here, the calculations were done with sort of our vanilla 4 angstrom live set around all the ligands, 6 angstrom real set. We did keep a flap order, what's so-called the HIV protease flap order, which we knew was critical to binding for this set of compounds. These calculations took about 25 minutes per ligand on pure CPU and gave pretty good correlation with experiment as shown to the right. So what I want to move to now are 2 sets of more systematic benchmarks. One of them is based on this very nice benchmark protein-ligand binding set developed at Merck by Schindler and co-workers, as referenced at the top of the slide. This has 8 protein targets, each with 26 to 44 ligands of known affinities. This is their measurements. I'm not sure if they're all their measurements. Some may be from the literature. They give very nice protein setups, ligand starting conformations, which we used. But I should mention that we also have done these calculations using the map snap setup and got very similar results. In our initial calculations, we omitted all crystallographic waters, used a full continuum solvent representation, and again used this 4-6 sort of initial starting live real set that we find work pretty well as a starting point. The force field is fairly straightforward. We used amber tools to generate amber 14SB parameters for the protein, GAF2 non-bonded parameters, the ligands, except that we used our V-charge electronegativity equalization method to provide the ligand charges. It's quite a fast method and gives charges that are compatible with amber and charm, so forth. One of the things that's nice about this benchmark set is that Schindler and coworkers ran and provide results from Schrodinger's FEP plus technology, as well as 2 ways of using docking and scoring and other methods. So we can look at our results in comparison with these other approaches. The point of the way we set this up was to do a uniform setup for all the systems. So although this is retrospective, it's fairly systematic. This is a summary of the results. So we're plotting R, the correlation coefficient, for each confirmation calculation or set of calculations. And here you see results, a cluster of columns for each of the different protein targets. Each column is a different method. So in dark blue is our VM2 calculation. In yellow is the FEP plus. Gray is docking with glide, and this pinkish color is a different way of using the glide scoring method on the docked poses. And in green is the MMGBSA method in prime, also a Schrodinger product. And what you can see, so what we have over here is a weighted average over all the systems, where the weighting is by the number of ligands in each system, since that varies from target to target. So I think the overall result that I would call your attention to is that VM2 is more accurate than docking and prime MMGBSA. And if we do statistics, I think it's significant. It's not as accurate as FEP plus, which is perhaps expected given the simplicity of the method, but what that gets you, on the other hand, is a lot of speed. So these calculations take about 20 minutes per ligand. If you're running across 12 CPUs, you could get it down to about 7 minutes per ligand pretty easily on a GPU. So whereas the free energy type calculations are much more time consuming. Now, as I mentioned, these calculations were done with no crystallographic waters, no explicit waters at all. Let's see what happens if we include the crystallographic waters and keep them in place as we do the calculations using some weak restraints. We see overall is that there's not a lot of difference. So we go back and forth. Basically, the R value stays at about 0.5. Note that all the other calculations are the same. We're just changing VM2. But I mean, I guess one thing I should have pointed out is that there are some cases where VM2 is more accurate than FEP plus. These are cases where maybe a continuum solvent method is working well, because you can see MMGBSA working well. SHP2 is another example. I should also mention that, going back a little bit, that when we looked at the scatter plots of calculation versus experiment, there is one extreme outlier for the protein sick. And when we looked at what was going on, it turned out that actually the experimental data had been mistranscribed onto GitHub. So when we fixed that, the outlier went away. So that was sort of an encouraging example of the calculations helping us find a small error in the reported experimental data. Finally, there are different ways of running VM2. We could expand the real set. We could expand the live set and the real set. We could add explicit crystal waters. And so you can imagine a typical scenario, especially if you have some compounds with experimental data known, and you can use that to optimize how you do the runs. Here are results where we did giving the best results over these different small variations. And here you see, with a little bit of tuning, the calculations get even better, become more competitive with the more time-consuming FEP plus method. So to look for consistency, we then went ahead and did essentially the same study using what's called the open force field benchmark. This is available on GitHub, and mainly these are systems that were studied by Gapsis and coworkers using 3 different free energy approaches. So there are 12 protein targets. Each one has, I think, 8 to 42 ligands of known affinity. We're leaving out here those members of the open force field benchmark set that are in the Merck subset, in the Merck benchmark, which I just talked about to avoid repeats. We also are leaving out 2 metalloenzymes. But what's nice about this, again, is we can look for consistency of VM2 results across other systems and compare more extensively with various free energy methods. So FEP plus from Schrodinger and some non-equilibrium approaches to doing the free energy calculations with two different force fields, all put together in this paper from Gapsis and coworkers. So the interpretation of this graph is similar to what I showed you before. Here we have a cluster of columns for each of the protein systems with its own set of ligands and the different methods. So again, dark blue is VM2 with a sort of very simple initial starting run of 4-6 live real set. So 4 angstroms from the ligands, 6 angstroms from the ligands, and no crystallographic waters included explicitly. And then in the other colors, we have FEP plus as run by Gapsis and coworkers, and then these other two non-equilibrium methods in the other two colors. So again, the R value for VM2 is quite similar to what we saw for the Merck set, about 0.5. Again, it doesn't perform as accurately as the FEP plus or other two methods, although it's certainly closer to the other two methods, and in some cases performs exceedingly well. But again, the time is dramatically shorter. So this, again, points to this sort of distinctive balance or combination of accuracy and speed that I think makes Mining Minima quite an interesting new approach. We don't know what's going on with PTP1B. We think that this has to do with the high level of charge in the system, but we're looking at that now. Again, we can look at what happens if we include crystallographic waters explicitly. And for this set of proteins, the R value actually does go up somewhat for VM2, which is nice and seems intuitively reasonable. And again, we can look at what happens if we try a few different variations with different live real sets or adding the explicit waters and give the best VM2 results, which again points to the possibility of some minor tuning of the VM2 calculations with an initial set of ligands of known affinity that would improve the accuracy of the calculations going forward. I won't spend a lot of time on it, but VM2 also has given quite nice results for a set of host-guest systems spanning various chemical types, cyclodextrins, octaacids, and cucurbiturils. So we've been excited recently to be working with folks at the Chemical Computing Group, the creators of the Molecular Operating Environment, or MOE, software, to begin integrating VM2 into this very powerful GUI. So in this approach, there's another menu entry for VM2 to import its results. And just to give you a sense for how this works, this opens up a MOE database where each row corresponds to one ligand. And of course, there are multiple conformers for that ligand in the bound and free states where one can drill deeper, as I'll show in a moment. If there are experimental data in your data set, you can make this scatter plot of calculation versus experiment. And there's some really nice tools for drilling down on the results of this interface. In addition, if you then go to the results for one ligand, as shown in this row, which corresponds to one row in this table, you can open that up into another database and then explore more deeply into the conformers and energetics of that ligand in the protein and free in solution. And what you can begin to do is browse through ligand conformers and look at how the structure of the ligand varies in the binding site. So these are different conformers of the ligand that are shown simultaneously as generated by VM2. And you can begin to see how the protein binding site has relaxed differently in response to each of those conformers, which makes sense and is captured pretty well by this method. Another direction that we think is really interesting for VM2 and perhaps makes it a bit more unique is that it offers the capability of looking into the thermodynamic contributions that lead to either high or low affinity. So in addition to free energy, which is of course the main thing we're interested in, one can break out changes in energy and entropy, or that is the configurational entropy of the protein or ligand on binding, and then go deeper into energy terms like the electrostatic energy, solvation free energy, and so forth. And I think these kinds of analyses, along with breakdowns by structural components, so for example, how much does this part of the ligand contribute to the free energy? These are breakdowns which are somewhat heuristic, but I think can give insight that, again, can help guide subsequent stages of the design of high affinity ligands. I want to show you just one slide on this method that's in development but is operational now called Quantum VM2 or QMVM2. The basic idea here is that we use a divide and conquer method available in the open software games from Iowa State to treat the whole protein and the ligand quantum mechanically. The way this run works is that we start off with a classical VM2 run, just like the ones I've been talking about, to generate the conformers and get the entropies of each of the configurational entropies of each of the conformations. So this will be a ligand. What I'm going to show you actually are results, again, for the set of HIV protease inhibitors from University of Massachusetts. So again, we do a ligand 2D to 3D calculation with our vconf software. We use MapSnap to map each of these ligands onto the bound pose of the known crystal structure. And we use classical VM2 to get conformations and entropies of multiple poses. We then identify the 30 most stable conformers and run those through a more detailed QM calculation. So this is a divide and conquer semi-empirical method where the protein is broken down, is divided by residue. For each pose, we use the QM Hamiltonian or energy functions to relax the live set just as we would with a force field. And then that gives us a new and more refined QM energy. The QM calculations are done with a typical implicit solvent model PCM method. We combine these energies with the entropies from the molecular mechanics results. And here, at least in this initial study, the QMVM2 results actually improve the accuracy of the HIV protease calculations that I showed you before with an R-squared of 0.76. So I'll wrap up by summarizing some of the main points. I think VM2 offers a distinctive and valuable balance of speed and accuracy. And I think this opens it up for a number of applications. One natural one would be virtual screening after docking calculations. So if you dock, let's say, 100,000 compounds, you may want to run VM2 on the 1,000 most promising looking ones to get a more accurate readout of their likely affinities. I talked about the idea of using it in an interactive mode where one does ligand design in Mo, for example, and then shoots out suggested ligands in proposed poses to get feedback on them in a minute or so using FE process. And then this idea of doing a detailed analysis of what are the drivers of affinity in terms of energy, entropy, and molecular components. The VM2 software runs on workstations using several types of parallelism, including GPUs, and is available on AWS Cloud for surge requirements or for those who don't want to maintain their own hardware. And we're excited about these work in progress of integrating VM2 with MOE to make it more accessible to a wider range of users, and also the integration of games to do more accurate calculations where you don't have to even worry about having a force field for the final calculation, and which I think also will open up a range of applications for metalloproteins, which are pretty common in drug design. So we're actually quite happy that InCerebro, a new pharmaceutical company, has adopted VM2 as a core of its computational pipeline. So we're thrilled to be working with them. And finally, I want to acknowledge the folks who have made this all possible. The team from VeraChem, listed in the top row, the folks from Iowa State who are working with us to integrate VM2 and games under the leadership of Mark Gordon, and then the folks at CCG, Chris Williams and Human, who have enabled that work. Thanks so much.